Welcome to the deep dive. So, immunotherapy. It's been a game changer in cancer treatment, really offering hope. But, there's this persistent problem. For way too many patients, it just doesn't work as well as we'd hope. Sometimes the response isn't complete or it doesn't last.
Right. And today, we're getting into why that might be. We're exploring some really cutting-edge research that looks inside the tumor itself. It reveals this kind of hidden complexity, showing how some cancer cells are just masters of disguise, invisible to our immune defenses. We're diving deep into subclonal immune evasion in non-small cell lung cancer.
And our guide for this is a fantastic paper, same title, Subclonal Immune Evasion in Non-Small Cell Lung Cancer by Dijkstra and colleagues. It just came out in Cancer Cell. It's genuinely eye-opening stuff. Absolutely. So our mission today is to unpack the key insights from this study for you. We want to help you understand this whole immunotherapy resistance puzzle and maybe see what surprising new directions this research points towards for actually treating cancer better down the line. Okay, so how did Dijkstra's team actually get a look at this? Because it sounds incredibly complex. They developed this really innovative experimental setup, didn't they? Not just theory.
Exactly. It's quite brilliant, actually. They used patient-derived organoids. Think of them as mini tumors grown in the lab from actual patient samples. But the key was they took these organoids, grown from multiple different regions of the same tumor,
Okay.
and then they co-cultured them with the patient's own T cells, specifically the TILs, the tumor-infiltrating lymphocytes that were already in the tumor trying to fight it.
So a direct functional test. You can actually see the interaction.
Precisely. It lets you observe, in real time almost, how different parts of the tumor, different cell populations, actually stand up to the immune attack. And what's really fascinating, just the scale they achieved here.
Yeah.
From just 3 non-small cell lung cancer patients, they managed to isolate and functionally test 81 distinct clonal sublines.
81 from 3 people.
81, sourced from 11 different regions across those tumors. This gave them this incredible resolution, like looking at the tumor diversity almost cell by cell, or at least subclone by subclone, a really unique window.
Whoa, 81 subclones. That level of detail must have revealed things you just completely miss otherwise, right?
Oh, absolutely. It completely shifted the perspective. The study's main sort of headline finding is this. Individual cancer subclones, these distinct groups of cells within the same tumor, they vary intrinsically, inherently in their ability to evade immune cells.
Intrinsically. So it's not just about where they are in the tumor, but something about the cells themselves.
Exactly. It means not all cancer cells, even from the same patient, are created equal when facing the immune system. You can have some subclones that are quite vulnerable, easily recognized and killed by T cells, living right next door to other subclones that are basically invisible, masters of evasion.
So hang on. It's not the whole tumor acting as one unit against the immune system. It's like different factions within the tumor, some hiding, some not. That makes treating it way harder, surely.
Precisely. That's the core challenge this highlights. And they found these differences weren't random. They used whole exome sequencing, RNA-seq, to show these variations are linked to distinct genetic pathways, different transcriptional programs. You could trace their lineage.
Okay.
And yeah, the crucial observation, these immune-evading subclones can happily coexist with immune-sensitive ones. In the same tumor region, even. It's like having soldiers with standard camouflage mixed in with soldiers who have, actual invisibility cloaks.
Right. And the ways they evade, the mechanisms. The study mentioned both antigen-dependent and independent methods, so multiple tricks up their sleeve.
Yes, it's not just one mechanism. Some might hide the flags, antigens that T cells look for. Others might put up do not disturb signals or resist the killing process itself. But this leads to maybe the most profound insight, I think, that this ability to evade seems to be a cell-autonomous property.
Cell-autonomous, meaning it's built into that specific subclone, like it's part of its own programming.
Exactly. It's not primarily something they're learning from their neighbors or the environment. It's inherent to that particular line of cancer cells. It's hardwired.
Wow.
Which has major implications, right?
Yeah.
It suggests that maybe therapies need to be subclone specific, and it raises this big question. If these stealthy subclones are hiding among more vulnerable ones, how do you even find them, let alone target them specifically?
Yeah, that's the challenge. But this study really seems to push the field forward. It sounds like the first time anyone actually shown this functionally, right? Directly demonstrated it.
That's its major strength, yes. The first direct functional proof that these genetically distinct subclones, same tumor, have fundamentally different capacities for immune evasion built in. We're seeing it happen in the dish.
Not just inferring it from sequencing data.
Correct. And beyond just the finding itself, the method is huge. They've established a scalable way to actually screen tumor subclones for immune evasion. This moves us way beyond just describing the genetic landscape. We can now link specific genes or pathways to the function of evasion.
So it connects the dots between tumor evolution and immunology.
Beautifully put.
Yeah.
Yes. It shows how that relentless process of subclonal evolution within the tumor directly shapes its ability to hide from the immune system.
And the clinical relevance is just staring you in the face then. This gives us real mechanistic clues about why some non-small cell lung cancer patients just don't respond well to immunotherapy.
Absolutely. It immediately suggests we need strategies that can account for this heterogeneity, maybe ways to unmask or specifically target those evasive subclones.
Okay, but like any study, especially one this innovative, there must be limitations, things to keep in mind.
Oh, for sure. And the authors are upfront about them. First, the patient cohort for the deep dive, the organoid work, was small, just 3 patients. So while the findings are profound, we need to be cautious about generalizing immediately to all NSCLC patients. More work is needed there.
Right, 3 is a small sample size to draw broad conclusions from.
It is. And second, the model itself. These organoid T-cell co-cultures are powerful, much better than simple cell lines, but they still don't fully capture the entire complexity of a tumor in vivo. You're missing some of the other cells, the structural components, the signaling crosstalk of the full tumor microenvironment.
Makes sense. It's a model, not the real thing.
Exactly. And finally, while they linked evasion to distinct genetic lineages, they didn't definitively nail down the specific molecular drivers, the exact genes or mutations causing that evasion phenotype in every case. That's a key next step.
Okay, so that leads naturally to the next question. If this study opens these doors, what's the research roadmap look like now? Where does this field go next?
Well, several clear directions emerge. First, obviously, expand the patient cohorts. Look at more patients, different subtypes of NSCLC, maybe even other cancer types, to see how general these principles are.
Get broader validation.
Right. Second, longitudinal studies. Track tumors and their subclones over time, especially in patients receiving immunotherapy. How does the subclonal landscape change under treatment pressure? Do evasive clones get selected for?
See the evolution in action.
Exactly. Third, dive deeper into mechanisms. Pinpoint those specific molecular pathways, the genes, the mutations, that confer this cell-autonomous evasion. That's critical for finding targets.
Find the invisibility cloak genes.
Sort of, yeah. And fourth, based on that, therapeutic targeting. Can we develop drugs or strategies that specifically hit these immune-evading subclones, or maybe resensitize them?
Makes sense.
And finally, refine the models. Incorporate more elements of the tumor microenvironment into these co-culture systems to get an even more realistic picture.
So, quite a lot to do then.
Always in science.
Yeah.
But this study provides a fantastic launchpad.
Okay, so let's bring this all together. If there's one big aha moment from this deep dive, it really seems to be understanding just how incredibly diverse a single tumor can be. And that this diversity, right down at the subclonal level, is a major reason immunotherapy faces challenges. It's not fighting one enemy. It's like fighting a whole varied population.
That's it, exactly. Grasping this intrinsic cell-autonomous immune evasion, clone by clone, is fundamental. It's a critical step if we want to move towards treatments that are truly personalized and, hopefully, much more effective, connecting it to the bigger picture. It just underlines that cancer is incredibly adaptive, and our therapies need to become just as adaptive and maybe multi-targeted.
So here's a thought to leave you with. Imagine a future where cancer treatment isn't the sort of broad spectrum attack, but instead it's a highly targeted, maybe even dynamic strategy, one that specifically identifies and disarms the most elusive, the most immune-evading subclones within your unique tumor. This research suggests that, the map to that future is actually starting to be drawn one subclone at a time. Thank you for joining us on this deep dive. We hope you'll continue exploring the fascinating world of science with us.